And then I says to the guy, John, that's not a donut, that's my ex-wife. What? Oh, it's not? Oh, hello there, I'm Fahad, as Himo said my name. And then I says to the guy, John, that's not a donut. And then I says to the guy, John, that's not a donut, that's my ex-wife. What? Now? Oh, hello there, I'm Detective Fahad. I thank Himo for giving me this time, and also all this nice equipment and this camera. I'm just going to tell you a little bit more about DNA finger. I'm just going to tell you a little bit more about DNA fingerprinting. So, as you know, the names of DNA fingerprinting can be DNA fingerprinting. As you know, the name. Then I says to the guy, John, that's not a donut, that's my ex-wife. Now? Oh, sorry about that. Hi, I'm Detective Fahad, and I'm going to be telling you about DNA fingerprinting. I would just like to thank Himo for giving me this time and all this beautiful equipment to do this segment. So, back when I was a cadet, they would teach us all this stupid info, nobody needed to know it, but turns out I need to know it for now. So, as you know, there are other names for DNA fingerprinting: DNA typing, DNA profiling, genetic fingerprinting, genotyping, or identity testing. In genetics, method of isolation.
Today's story will present to you something about technology. By that we mean DNA fingerprinting. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene is compared with a DNA sample taken from a suspect. DNA fingerprinting is also used to establish stuff like paternity.
Aliza! Can you come here really quick? I need help. Are you coming?
What?
Okay, can you go around, make sure not to touch anything, go around.
Okay.
Do you see that black wire? Over there, near the plug, at the plug. At the plug. Go around. You see that black wire right there? In the plug.
Yeah.
Move the curtain. Okay, now push against the Wi-Fi thing. The big white thing, push against it. From the bottom. No, push against the Wi-Fi thing. You know, the thing with the green lights in it?
Yeah.
Push against it from the bottom. And now take out the black wire while you're doing it.
Is there a button at the part?
No, take out the black wire while you're doing it.
Okay.
Now give it to me. Close the door.
Yeah.
things that DNA fingerprinting are used for. Used in court to identify certain cases to track down bodies. Is also for medical uses. It is used for cures d'azur. As we can see, we see a lot of those test kits that you can buy and send off your DNA, and they will send you back a family tree or something of the sort. Another medical procedure that they have is used to match tissue of organ donors, those people who are in need of transplants. So a little bit about the history of DNA fingerprinting. It was a technique developed in 1984 by British geneticist Alec Jeffreys after he had noticed that certain sequences of highly variable DNA, which do not and these strands do not contribute to the function of the genes and are repeated in the genes. Another thing that they use for are DNA fingerprints, are used for chemical tests to identify genetic makeup of a person or a living thing. They also have other names such as DNA fingerprinting, also called DNA typing, DNA profiling, and genetic fingerprinting. What you have to do to get one of these. To get your DNA fingerprint, you have to send in a sample of yourself or your body, which can be a cotton swab going in the mouth, or the use of skin, a hair strand, or saliva, sweat, or any other body fluid. But most places that do this testing, usually they like to use blood because it's the easiest way. A way that this works is, so basically you give the sample, and then they extract the DNA, and then after that they purify the DNA, and then they do the polymerase chain reaction, and then that reaction takes place, and you get your DNA sequence chart, which looks something like this. Anyways, that's all for me. Off to Officer Fahad.
And then I says to the guy, John, that's not a donut, that's my ex-wife. What, that's no? Oh, sorry about that. Hi, I'm Detective Fahad, as Himo said, and I'm here to teach you about DNA fingerprinting. First off, I'd like to thank Himo for sending all this beautiful equipment and letting me do this segment. First,
What?
हेलो हेलो अम्मी है? मैं आ नहीं सकता, मैं एक ग्रुप प्रोजेक्ट अभी कर रहा हूँ, मैं अपना पार्ट कर रहा हूँ फिल्म करना है। अच्छा Get the trolley and go downstairs. She I don't like it. And then I says to the guy, John, that's not a donut, that's my ex-wife. That's now, oh, hi there, sorry about that. I'm Officer Fahad, as Himel said, and I'm going to teach you about DNA fingerprinting. First off, I'd like to start off by thanking Himo for sending all this beautiful equipment. Now, let's get started. The technique of DNA fingerprinting changed the era of identification, characterization, and the classification of organisms. Notably, approximately 99% of our DNA is similar. A 0.1% difference is sufficient to make someone so unique. We are using this 0.0%. Notably, approximately 99% of our DNA is similar. A 0.1% difference is sufficient to make someone unique. We are using this 0.1% portion of DNA profiling, which is often known as DNA fingerprinting. This is the reason why only DNA is used for DNA fingerprinting, since it's in the name, and in all body parts, tissues, and cells, except germ cells, our DNA is the same. Even after death, the individual remains the same. It can remain Notably, approximately 99% of our DNA is similar. A 0.1% difference is sufficient to make someone so unique, and you 0.1% beautiful. We are using this 0.1% portion for DNA profiling, which is often known as DNA fingerprinting. In all our body parts, tissues, and cells, except germ cells, our DNA is the same. Even after death, the individual still has its. In all our bodily parts, tissues, and cells, except germ cells, our DNA is the same. Even after death of an individual, it remains the same. It can remain stable for even after 1,000 years. That's how they identified King Tut. In all our bodily parts, tissues, and cells, except germs, our DNA is the same. Even after the death of an individual, it remains the same. Our DNA can remain stable for up to 1,000 years. As we all know, 97% of our genome is non-coding. Now, 97% of our genome is non-coding, repetitive, and junk. Protein is formed. 97% of our genome is non-coding, repetitive, and junk. Protein formation is regulated by only 3% portion. We know it as genes. Moreover, repetitive nature and polymorphic proteins of junk DNA are utilized to perform DNA fingerprinting. The number of repeats and the sequence structure of those regions varies between individuals and organisms based on the entire. Moreover, the repetitive nature and polymorphic properties of junk DNA are utilized to perform DNA fingerprinting. The number of repeats and sequence structure of those regions varies between individuals and organisms based on that the entire DNA profile or the DNA print can be prepared. Now, let me give you a comprehensive overview of the whole thing, Now, let me give you a comprehensive overview of the whole method down below. Now, let me give you a comprehensive overview of the whole method. The DNA is isolated from a bodily sample. As said before, this can be only anything that comes from the body: sweat, saliva, hair, skin, mostly anything. Digested or amplified use. Now first, the DNA is isolated from a bodily sample. As we know, this can come from anything that comes from our body: our hair, our saliva, our cheeks, your sweat. This sample is digested or amplified using restriction digestion, or PCR. More on that later, respectively. The DNA is isolated from a bodily sample. As we know, this can be from sweat, hair, saliva, almost anything from the body. Now, this sample is digested or amplified using restriction DNA. The DNA is isolated from the bodily sample. Now, as we know, this bodily sample can be almost anything from the body, a few examples being hair, saliva, and sweat. This sample is digested or amplified using restriction digestion, or PCR. More on that later. Using either agarose gel, This is done using either agarose gel, electrophoresis, or sequencing. The DNA fragments are separated and identified. PCR is shorthand for a very simple but very useful procedure in molecular biology called the polymerase chain reaction. It is a technique used to amplify a segment of DNA of interest or produce lots. Now back to PCR. PCR is short. Now back to PCR. PCR is shorthand for a very simple yet useful procedure in molecular biology called polymerase chain reaction. It is a technique used to amplify a segment of DNA of interest or produce lots and lots of copies. In other words, PCR enables you to produce millions of copies of DNA sequence from an initially small sample, sometimes even a single copy. It is a crucial process for a range of genetic technologies, and in fact has enabled the development of a suite of new technologies.